I'm Glenda Gray. I'm the director of the Infectious Disease and Oncology Research Institute, and we host these seminars where we bring wonderfully acclaimed people to come and talk to us. And we're very lucky to have Simon Waddington and his son join us for the seminar. And Simon began his journey in preclinical gene therapy. In cystic fibrosis and Duchenne muscular dystrophy, and then hemophilia. And so he's going to give us a lecture on gene therapy and some of the work that he's been hosting. And I just wanted to say thank you, Patrick. I think Patrick's and his group of the experts, in the local experts, so it's wonderful to have you here, join us. And afterwards we'll have question and answers. And then we like to do, because we have students with us, it's always nice to do we have a networking after this where we have some food and some wine.
Great.
And we can then talk further and gives an opportunity, especially for students, to interact with you.
Sure.
So thank you so much for
joining us. Thank you very much indeed. Thank you. As per my introduction, I started working in gene therapy in I kind of was doing it kind of part-time from about 1996 onwards, but then joined formally in 1999, and initially it was cystic fibrosis, which is a really hard target and probably not the best target, and then moved on to hemophilia. Had a great time actually working with people in hemophilia, and then moved on to more severe early onset childhood neurodegenerative diseases. Had some interesting work there and some good success. And as per gene therapy, the early gene therapy successes were for the more easily treatable diseases. And so as we've progressed, we've targeted more difficult to treat diseases, and also because the technology has improved so vastly, it can now be applied to a whole bunch of different diseases. Now, the way this talk I'm going to present it is that I'm going to talk about some extremely basic concepts that probably almost all of you understand. I'm not sure whether my son Zane understands this or not, because I'm not sure whether you did A-level biology. But anyway, but it's really basic concepts, but I actually will also add discussions that are relevant to the concepts of gene therapy. I'm going to talk about how viruses infect us, which is really important for viral gene therapy. And I appreciate that viral and non-viral gene therapy now are almost acquiring equal footing across the world because of the technological improvements in non-viral genetic therapy. And then I'm going to talk about how we've exploited viruses to create viral vectors. But similarly, actually, is that we're now creating virus-like particles where we've used our learnings in viral gene therapy to create non-viral constructs. And then I'm going to move on to what Hollywood makes of it all. And this is kind of the fun bit, where I'm going to show you a bunch of films that discuss gene therapy, and then I'd like you to guess the films if you can. And then I'm going to discuss how Hollywood got it wrong and what the actual truth is of the matter. Okay, so I'm going to start with this here. And this is Amanda Valerina Cardoz. And she had type 1 spinal muscular atrophy, which is a devastating inherited genetic disease which affects the motor neurons. And so this is her at 18 months. Cognitively, she's capable, but in terms of her motility, she was on ventilation, and the usual situation with infants with type 1 spinal muscular atrophy was that they would rarely survive beyond their second or third birthdays. This is the sort of disease that the type of disease class that I've been looking to treat over the past 10 years. And I'll loop back to this disease towards the end of my talk. Okay, so just to give you a little bit of interesting historical context, this is Edward Tatum, who won the Nobel Prize for the one gene, one enzyme hypothesis. And he was asked to write a horizon scanning piece in 1966 about where he thought biology was going. And he said that finally it can be anticipated that viruses will be effectively used for man's benefit in theoretical studies, in somatic cell genetics, and possibly in genetic therapy. So this was incredible foresight, because in 1966, this was 4 years before the dawn of recombinant DNA genetic engineering. So this is 4 years before people discovered the application of restriction enzymes, 4 years before people realized that you could amplify bacterial plasmids in bacterial cultures. So it was pretty far-sighted. So this is the simple bit that we start off with here now. As I'm sure most of you or all of you are aware, every cell in a body contains a nucleus, and the nucleus encodes RNA, which is transcribed into proteins and then decoded by ribosomes. This is the general simple tenet, and there'll be a reason why I'm saying such simple stuff in a minute, of course. So in a genetic disease, in some genetic diseases, there's a mutation in DNA which results in the synthesis of a mutant protein, or sometimes no protein at all. So here what you're seeing is, for example, severe hemophilia B, where you've got a stop codon in that caused by that mutation. The ribosomes stall on the RNA, so you never get that protein produced. Of course, genetic mutations are far more complex and diverse than this. The simplest diseases that were treated by gene therapy were this sort of disease, where it was a recessive genetic disease, whereby the patient inherited 2 recessive copies, didn't make any of the protein, and therefore, with gene therapy, if you just got back some of that genetic material, got the cells to make a little bit more protein, you could treat or even cure that genetic disease. However, there are more complex genetic diseases, such as haploinsufficiencies, where just missing 1 copy of that gene is enough to cause the genetic disease. And one of the diseases that we work on is an inherited epilepsy called Dravet syndrome, which is caused by a mutation in one of those alleles. As a further twist to this, of course, you might have a genetic disease where that mutation causes a dominant negative effect. And therefore, for gene therapy, you'd have to knock out that mutant copy as well as putting back a working copy. So as time has gone on, genetic therapies have been applied to harder and more complex types of mutation. And here is a simple list of genetic diseases and their mutations. This was an early list when gene therapy was in its early development. Now, of course, it can be applied to a whole bunch of diseases, and I'll be going into some of those diseases when I talk about the movies. So now, just switching discussion a bit. Here we have a virus about to dock with a cell surface protein. So viruses have co-evolved with eukaryotes for about 500 million years. And so here we have a cell with its natural receptors or transporters, and these viruses have evolved to hijack one or more of those receptors, hijack the intracellular transport machinery to be able to transport themselves into the nucleus, which is what this one is doing here now. So it's actually using the hijacked cellular machinery to transport its genetic material into the nucleus. It then is hijacking the replication machinery to make huge numbers of copies of RNA in order to be turned into viral proteins, which can then be released into the bloodstream, for example, in quadrillions of copies. In gene therapy, for viral gene therapy, we have learned to use to redesign viruses so they can be used as vectors to deliver our genetic material. So on the left here, we have a picture of a virus. On the right here, we have a picture of a gene therapy vector. And the point I'm making here is that they look the same on the outside. They look the same on the outside to us, and they look the same on the outside to a cell. But on the inside is the viral DNA that contains all the genetic material to encode the proteins to deliver and produce more copies of that virus, whereas in gene therapy, we're just delivering the DNA that we want to deliver. Plus, at the very ends, you've got a little packaging signal that allows it to be stuffed into what looks like a viral particle on the outside. So here we've got, again, going back to our genetic disease, this cell cannot make that protein, and you've got our vector, which looks like a virus on the outside, contains copies of those genes to be delivered into the cell. It then recognizes the same receptors, hijacks the same cellular machinery, but in this case delivers working copies of the genetic material, which means this cell can now make protein copies for the first time. This has relevance that I'll mention later, because to us and to the cell, these things look like viruses, which means that there is immunity to gene therapy viral vectors in the same way there is immunity to viruses. And we have really good antiviral immune responses, as well as adaptive immune responses. So this has been one of the fairly major hurdles with viral gene therapy, which is why non-viral gene therapy has always been a second horse in this race, one where it doesn't contain viral proteins and therefore might not evoke immune response, but generally hasn't been efficient. But because of the cross-pollination of the knowledge between these two almost parallel fields, one which is a top-down approach and one which is a bottom-up approach, you've now had learning from both fields to create genetic delivery systems which can be vastly more efficient than we imagined they would be 20 years ago. Okay, we're now going to move to Hollywood, and I'm going to show a bunch of films, I think in reverse order, and I'd like you to try to guess the films, and then I'll just discuss the points that one might learn from these. Hopefully the sound works. Any idea what the film is? But no guesses. No guesses? Sorry? What? It is The Rock, yeah? Sorry? It is Rampage. Okay. where there's a genetic modification of a gorilla. So if we believe Hollywood, obviously The Rock knows what CRISPR is, which seems pretty unlikely, and also that growth, strength, and speed are determined by a single gene. But there's another point here, and this is, of course, to do with CRISPR. So I think that sometimes people have conveyed the idea that CRISPR and other editing technologies are kind of like the next evolution in gene therapy. Because, you know, the gene therapy for hemophilia is a kind of rather brute force gene therapy where you're just putting in extra copies of the gene. So it's a bit like a kidney transplant. In a kidney transplant, you don't take out the non-working kidneys. You just put in an extra kidney, and you don't even put it in the same place where the other kidneys are. You kind of plumb it in down there. This is a bit like kind of old conventional gene therapy. CRISPR is a way of actually editing the genetic material in situ, editing single bases or stretches of DNA. And this is a beautiful approach and has been used in numerous mutations in genetic diseases. The problem is that because a genetic disease may be caused by a range of different mutations in any part of that gene, and because CRISPR is designed to target a single region of that genetic material, it means that you have to design a guide against each of those different mutations. And that's a lot of work. So therefore, CRISPR isn't necessarily a cure-all for all genetic mutations. We're still going to use the old-fashioned gene therapy for a lot of diseases. But CRISPR allows a level of tuning of finesse. Essentially, CRISPR, base editing, prime editing, other types of editing, they just expand the the toolbox that we've got to be able to treat a range of genetic diseases. Okay, next film. Any idea of the film? Any idea of the actors? Yes, Jared Leto. Right. Okay. Good. At least you got the actor. Okay. Okay. Does anyone do cloning in this room, by the way? Yes. Okay. Right. So I'm just going to rewind that, and then you. Oh, well, actually, no. You can look at it. You can see it here. If you look at that, that's the amount of time it took. I think that's 13 hours, 16 minutes, and 30 seconds to clone some bat DNA into human cells. Do you think that's a reasonable representation of cloning? So, as we all know, it sometimes takes a really long time. You might get the cloning to work first time, but generally, cloning will take days, possibly weeks, and sometimes, in our experience, when trying to develop gene therapy vectors, it can take months, or you just give up and you just try to get it synthesized from scratch. In other words, it is a long journey, and developing these things from first concept all the way to a clinical trial is a lot of work. I mean, it requires focus, teams, and dedication, and the depth of experience to be able to get these things all the way from the bench to the bedside. Okay, next film.
What he's saying is great. Just start at the beginning. Born to British parents, and then at 19, you discovered a gene therapy that literally doubles the life expectancy of pancreatic cancer. Actually, it tripled their life expectancy. But it's okay.
Any idea of this film? You're not so good at films, are you? That's okay. Sorry? No, it's not. No. That's Tom Hardy. Tom Hardy, who is in what? He's in this film. Here, there we are. He's in Venom. So the point here about this movie is that I've been talking about gene therapy for inherited genetic diseases, and so the field I work in is where you are delivering vectors that will actually contain therapeutic stretches of genetic material to treat a genetic disease, and they need to get in as stealthily as possible to avoid an immune response. Because you have an immune response against your vector, that means the body will eliminate your vector and your therapy doesn't work. However, the field of genetic therapy is huge, and it also encompasses genetic therapy for cancer. And I would say there are two main branches of cancer gene therapy that are currently showing a huge amount of promise. The first is CAR T cells. This is where you take, in some cases, a patient's CAR T cells out, you genetically modify them using a lentiviral vector or using CRISPR, to redesign them so they are targeting the patient's tumor, and then you re-inject them into the patient. In other words, you're redesigning their immune system to target their tumors. The second major cancer gene therapy is oncolytic virotherapy. And this is where you're taking viruses that have been disabled, but they will deliver toxic genes. And these viruses, they have been tweaked so they target tumors, and they will create an immune response, and they will create inflammation, and their toxic genes will kill cancer cells. So in effect, they are almost diametrically opposite to the sort of vectors that we design in our field. However, we're using a lot of similar tools, and I just want to therefore convey that gene therapy isn't just one thing. It's many, many things. And one of the things that I will talk about at the very end is gene therapy to reprogram cells to make monoclonal antibodies. Anyway, next film. No idea? Yes. Very good. Which Planet of the Apes? Sorry? Bang on. Absolutely. That's right. Yes. Yes, it is. Here we are. This is... Anyway, I'm not going to spoil it for you, but it's... So, if Hollywood is to be believed, gene therapy is passed on to your offspring. According to... Sorry, if Hollywood is to be believed. So, as far as I understand, although it's a very long time since I've watched this film, if I ever did at all, I'm not sure. And so the idea, I believe, is that Caesar was born from a chimpanzee that received the gene therapy. So actually, he inherited the genetic material from his mum. Certainly across the world right now, there is an international prohibition, a very serious moratorium on the concept of germline gene therapy. And in fact, when gene therapy is performed, there are rigorous screens for the germ cells to make sure that the gene therapy is not passed on. In other words, we're not actually modifying the human race. And I remember, and of course you might remember the Chinese scientist He Jiankyu, who actually did this, editing two embryos to develop HIV resistance. Anyway, so this is not the sort of gene therapy that we work on. Now, I mean, you know, there are interesting ethical considerations now, because now gene therapy is becoming so efficient that there are now thousands of children walking around that carry two recessive alleles for a lethal genetic disease for which they would have died years ago. And so therefore, are we now increasing the genetic load of extremely deleterious mutations? Anyway, this is not the point of this talk. The next point, if you believe Hollywood, is that it is so potent that just sniffing it as someone goes around with an aerosol is enough to transform every cell in your body, including your brain. As I keep on going on about, we are really good. We have really good antiviral mechanisms. And so when you aerosolize a vector, it will hit your lungs. Your lungs have got cilia that beat the vector out and away from your airways. And also we have really good antiviral mechanisms in our lungs as well. So it's been really, really difficult to efficiently deliver genetic material to the right cells. And certainly it's been really difficult to deliver genetic material, for example, to the central nervous system. And it's only in the past 10 years that huge strides have been made in developing vectors that are able, for example, to cross the blood-brain barrier to deliver genetic material to the brain for treatment of, for example, neurodegenerative diseases. The other thing, if Hollywood is to be believed, is that it will make you super intelligent, but only if you're an ape. Okay, right. Nearly the next, one of the final films. Second to last film. No. Any idea of the film? Sorry? Yes, yes. Yeah, you can come again. You, yeah. So I Am Legend. Yeah. So this is Will Smith with his. Anyway, kind of goes bad for Will Smith's character here. So, okay. So if Hollywood is believed, gene therapy vectors often turn into super viruses, in converting everyone in the world into flesh-eating zombies. So the point I like to convey with this here is that viruses, simply by virtue of their size, they can only contain a very restricted amount of genetic material. And for many viruses, there is so little genetic material that that payload can carry that they depend upon other viruses to help with their own replication. So in genetic therapy, in gene therapy, when we are manipulating viruses and trying to make them into viral vectors, inevitably, if we introduce any gene, it will destroy other genes in that virus. And viruses are so parsimonious with their genetic material that in many cases, a single stretch of DNA doesn't just encode one gene, but actually there are 2 different or 3 different reading frames that are producing 2 or 3 different proteins out of frame with the same genetic material, which means that inevitably we break viruses when we genetically manipulate them. And as we know from coronaviruses actually have been pretty good at generating their own mutations. Because the exciting thing about viruses, of course, is that not only do they have quite often quite poor replication that introduces lots of mutations, but it has now been, of course, shown in many instances is that if several viruses infect the same cell, they can actually swap each other's genetic material and help each other replicate. So generally is that, well, always, we break a virus if we try to think about genetically modifying it. This is the second to the last film. Any idea of the film? It's pretty hard, is this one, because it's quite a long time ago. Okay, I'm going to show you another picture, and you might get it then. Sorry? No, it's not Catwoman. No. And yes. Who guessed James Bond? Yes. Yes. It is James Bond. That's right. Yes. That short stretch with Pierce Brosnan. That's right. Yes. Yeah. I mean, one thing to learn is that if you ever see a film where there's spinning DNA in the background, you know it's bad news, don't you? So, and I'm not talking about the signs on the biopark outside here. This is, yeah. So apparently, according to Hollywood, ex vivo gene therapy is where bone marrow is taken from orphans, strays, and runaways to be injected into the patient and is known as DNA replacement therapy. Now, obviously, there's a few things to unpick here. So the first is that in terms of gene therapy for a range of diseases, but for example for primary immune deficiency. So you've got gene therapy where you are injecting a vector directly into the person, whether it be in their brain, bloodstream, airways, subretinally, wherever. So it's a direct gene therapy. Or what you do is that you take stem cells out, you genetically modify them, and then you put them back again. So the first is in vivo gene therapy. The second is ex vivo gene therapy because you're doing it outside of the patient's body. And then we move on to the point about the orphans, strays, and runaways. So people that receive ex vivo gene therapy, for example, for primary immune deficiencies, the conventional way of treating them is that they would be isolated in a bubble to prevent them from catching an infection because they were unable to fight off infections. And they would then go on a list to wait for a matched donor transplant. And so they would have a matched transplant. And a significant proportion of these kids that received a matched donor transplant, they developed graft versus host disease and died of the graft versus host disease because the donor's immune system had attacked their body. The point of ex vivo gene therapy is that the patient's own immune system is being taken out. It is being corrected and then put back in again. And it's been shown that infants that receive gene therapy have a better outcome than donor transplants because none of them can get graft versus host disease because they get their own immune system back again. Final film. Now, no one gets, well, only one person ever has guessed this when I've presented this. Yes, that's good, yeah. Yeah, but my God, 2 people have now got this ever now. Brilliant, yes, okay, yeah. The Bourne Legacy, yeah. I'm really impressed by that, okay. Bourne Legacy, okay. Now this one wins a gold star because Rachel Weisz's character, the first thing that she says is getting the gene where you want it is the nightmare. This is still a problem in gene therapy for a couple of reasons. The first is that vectors, whether they be viral or non-viral, you need to deliver them to what you think is the tissue that is the tissue that is primarily diseased. And as I referred to before, gene therapy for neurological disease has been incredibly difficult because it's been really difficult to deliver efficiently to the nervous system. It's still difficult to deliver to kidney cells specifically. And what you might want to do is you might want to deliver to where the tissue is most affected, but you don't want to be delivering that gene to tissues that don't express that gene because that might be toxic in itself. So this is a problem. One is that we're still on the foothills of trying to design gene therapy vectors that are targeting the right tissues. The other interesting thing is that now that gene therapy is becoming a thing that people are getting, we're now starting to realize that in some diseases that we thought that the muscles were, in Duchenne muscular dystrophy, it was always thought that the mainly affected tissue was the muscle, which is true. But it is now realized that the cerebellum is also affected by muscular dystrophy. So really what we need to be doing is designing vectors that not only target the muscle in this disease, but also target the cerebellum too. So we've got a lot of progress, and we are making a lot of progress with non-viral and viral vectors, but there's a lot of work to be done. The second thing that is really interesting is that her character says we're still struggling to put together a viral receptor map. Now you'd think that now that we've been using, in fact, I mean in clinical trials for 2 decades, more than 2 decades, and now market approved gene therapies for example, using adeno-associated virus vector, you'd think that we knew these vectors and knew exactly how they worked. We're still discovering the biological mechanisms by which they deliver their genetic material into the cells. So just imagine what would happen if we really had a good understanding of how what these mechanisms were, how much better we would be at delivering this genetic material. So all I'm saying here is that we've got a huge amount more to learn. We are seeing lots of successes now, but there is so much more to learn. Okay, I'm now just going to switch back to spinal muscular atrophy. So as you remember, Amanda Valerina Cardoz. So those videos there, they were uploaded by her mum when this must have been 10, 15 years ago, when there was no treatment for spinal muscular atrophy. So it's inherited neuromuscular disease, motor neurons die, infants will never sit unassisted or walk and will rarely reach their 2nd birthday. And for these infants, palliative care was the only possible care. There was no alternative. And this here is, on the left, is a natural history study of spinal muscular atrophy type 1. So on the left here, you've got the Children's Hospital of Philadelphia infant test of neuromuscular disorders. So it's an aggregate motor score of their performance over time. And age is along the bottom. And for this study, they actually see just a terminal decline because palliative care is the only thing they can do. And one child is still living out here at, I guess, 8 years of age, but very few of them live beyond 3 or 4 years of age. And as you can see from this review on the right, after 2 years of age, they generally only survive with a gastrostomy and a tracheostomy and non-invasive vest ventilation for more than 16 hours. It's a pretty grim situation. So in 2009, it was realized that an AV9 vector was able to, when injected early in life into mice or into non-human primates, could actually enter the peripheral nervous system. so several studies were then performed by different groups where they use an AAV9 vector delivered intravenously into a mouse model of spinal muscular atrophy. And in the mouse model, the mice die at 14 days of age, but when they receive the gene therapy vector on the day of birth, they survived for 2 or 3 months or more. So it was the first time that anything so transformative had been seen in spinal muscular atrophy. This then proceeded to a clinical trial in which 15 infants were recruited, and they were injected. They received their injections between, I think, 1 and I think it was 8 months of age. The design of this trial is pretty tough because what you're doing here is you're giving an injection of a gene therapy vector, one of which has never been tried in human beings before, and so it isn't the same sort of. You can't just do a big safety trial. Because otherwise you're immunizing a whole bunch of children against the vector that might cure them. You don't want to slightly cure them and therefore convert an early lethal disease into a chronic, horrible, long-lasting disease. And also you want to give every child the best chance of treatment. So the way this trial was designed was 3 infants would receive the moderate dose, and then if that was deemed to be safe, then the remaining 12 children would receive the higher dose. The first 3 children received the moderate dose, and so I'm now going to show you the CHOP-INTEND score of the 12 children who received the higher dose. And so here you can see that almost all of them achieved the highest CHOP-INTEND score that had actually been measured from the natural history study. 2 of the children here at the top here actually are above 60, which means they are indistinguishable from unaffected children. One of the children actually only plateaued, and this is possibly because that child received the injection when they were relatively old, and therefore it wasn't as effective. Possibly because at that age the vector was not able to enter the motor neurons, or possibly because at that point the disease had progressed to the point where you couldn't treat it as well. This is one of the patients at the top here. This is Evelyn.
Now 3-year-old Evelyn challenges Dr. Mandel to a dance-off at her annual follow-up appointment.
She comes back after 3 years, and she runs up to me and hugs me and says, Dr. Mandel, I love you.
I forget that she has SMA. This is a happy girl, and she does everything that a normal 3-year-old child would do.
So incredibly transformative, and that was her 5 years ago, and she's still fine. She's still doing well. So obviously is that there have now been a huge number of gene therapies that have received market approval, to the point now where actually I can't document them all because there's another one receiving market approval every day. The problem with spinal muscular atrophy is that Zolgensma, which is marketed by Novartis, is being costed at a single injection of $1 to $3 million for a single injection, which means that many healthcare systems across the world, this is impossible. So therefore, this is a very recent paper out in August 2025 in medRxiv, and this is from the range of countries that you see below. And they have been developing essentially what is a biosimilar for that AAV gene therapy, using an AAV9 again, using a different vector construct. And I'll just show you here, just summarizing there. They recognize that essentially this here is priced at $2 million US, severely limiting its access in LMICs. And so therefore, this vector, Versemnogene lantiparvovec, will be priced close to its manufacturing costs, and the tech will be transferred at 0 to minimal cost to individual LMICs. So now at least there is now rolling out the wherewithal to be able to start to design these things where they are, and they don't say what price that will be, so I don't know what it will be, but of course it will be vastly lower than what Zolgensma will be priced. The final paper I want to show this one here, and this was published online 21st of October. This, I think, is very interesting because it conveys a couple of points. So the first thing is, this is using a method of gene delivery called electroporation, I think. This is electroporation, is this one. And what this allows is reprogramming of muscle cells to deliver it to express a suite of monoclonal antibodies, which then produce an immunity against SARS-CoV-2. And what is interesting about this is that, as you can see here, it's a cold chain independent platform. It's much less expensive than other methods. And imagine the ability to be able to essentially create the wherewithal to produce an antibody suite. Now that could be used, of course, to treat or prevent an infectious disease. But of course, these antibodies could be used, and they have been used. These antibodies could be used to block an enzyme. They could be used to block a pathway in a cancer. And so the point that I think is so exciting here is this again is just another arm, another tool in the toolkit to be able to treat a whole bunch of genetic diseases that I think we're going to start to see rolling out across the world. Now the world is a lot more comfortable with genetic therapy. And of course, so many people across the world received genetic therapy when they were vaccinated against coronavirus, whether it be RNA or whether it be adenovirus vectors. Anyway, I think I'm going to wrap up there. I thank you for your attention, and I welcome any questions. Thank you.
Okay, so we're going to open it up for some questions from the audience. I'm going to ask the first question. So you showed that huge price, $2.1 million US.
Yeah.
So you told us that it's a simple viral vector.
Mm-hmm.
You stick some immunogen in, and can you tell me why it was priced at that amount?
Yeah. Okay, so I guess there's a couple of aspects to that question. So the first is that making vectors to GMP quality is expensive because the paperwork and the chain of documentation for every single thing that goes into that GMP product is incredibly expensive. And I now am becoming aware of FDA approval for GMP-like vector, which is greatly less cheaper less expensive to produce. So that's an actual cost that is that contributes to this. But there is another thing as well, is that the way the pharmaceutical companies are justifying this is that they would say, well, instead, you're not giving someone a drug that they will receive every month for their year, that therefore would cost a certain amount of money. Therefore, they are back calculating to say, this is a single dose that this person receive just the once that would treat them forever. Now, how this converts into a treatment for spinal muscular atrophy, where there is actually no treatment whatsoever, I don't understand that calculation either. But I do know that both these factors come into play.
That sounds perverse.
Yes.
But I'm going to ask
It is
Patrick
perverse, yeah.
about that. So I can understand the issue of making GMP grade material, because obviously one worries about safety and efficacy. So Patrick, maybe you could, so I know you got your own questions, but maybe we can take the issue around the cost of goods. And whether these interventions are completely disproportional to what they actually cost to make.
Well, Simon is correct to say that the GMP standard is quite a thing to meet, and that is very, very costly. And it's something that can't be trivialized, because if something is to be administered to humans, and it has to be of the absolute highest standard possible. So it's something that's very difficult to get around. But possibility of using GMP-like materials is one way that one could perhaps do that. But I think, what we are doing, and I think many people here are aware of things like the biorenewable ionizable lipids that we're using to deliver mRNA vaccines, which are based on using agricultural waste products. So I think that people are also focusing a lot on trying to generate more cost-effective or more affordable treatments for these serious diseases. So I think, this is not the end of the story using AAV9. There probably are going to be many more therapies that come out in the coming years that will be more affordable. But it's difficult to anticipate.
And I think that final paper where they were using the electroporation to deliver genetic material directly, it kind of speaks a little bit to what you say there. That there is so viral vectors are advanced therapeutic medicinal products, whereas delivering genetic material that's been synthesized chemically with associated chemical compounds is actually categorized differently, and therefore has different regulations, but it tends to be less expensive and therefore more amenable. And so I think what's happened is what I think actually has happened is that the success in the viral vector gene therapy and the teachings in this have now kind of started to instruct the development of non-viral genetic therapies, but also to make regulators and people more comfortable in the idea that this is a successful and a viable technology. So,
yeah, that's an important point. The viral vectors are quite complex to produce, a lot more complex than synthetic versions, such as using mRNA and non-viral lipid nanoparticles.
Yes.
Thanks for that wonderful talk, Simon. Was just interested about the article you mentioned where they try to make a biosimilar in low to middle income countries. And I was just wondering about issues around IP and how they're able to create that biosimilar.
Yes, I'm not sure. I mean, I know that they've used a different promoter to drive the expression of the gene, and I believe they've also used so when you express, I'm sure most of you know this, but when you express a protein because of third base wobble, you can actually different sequences that vary by the third base in the codon can produce the same protein. So they may have also designed the coding of the gene slightly separately, so therefore it uses a different bases, obviously to produce the same protein, and thereby stepping outside of the patent restriction the patent that they used for that exact sequence. So I suspect that's a couple of things, and also all the other bits that go into the vector, I think they've changed those as well. So it's just a variety of tweaks, probably just to move away from the IP of Zolgensma, I suspect.
So Simon, I want to ask you about that one baby. So you showed that slide, and there was that kid who got the therapy late and didn't develop enough immune response. You know you showed the high dose.
Oh yes, that's right. Yes,
So my question is around that was also a viral vector gene therapy.
Yes.
So could this have been because of so basically cross reaction? So this child already had an adeno-associated infection, and so when you gave the therapy, could it have been? Could it be because of pre-existing immunity to the adeno-associated virus?
So this cohort of kids, they all got huge amounts of immune suppression, vast amounts of immune suppression. And so it's possible, but it's not super likely, because they basically didn't progress much, and then kind of went they didn't it's pretty slow progression even then. But that brings me on to another point, and so that there have now been probably several thousands of kids that have now received Zolgensma. Enough that there is now a pretty large amount of data coming back in as to not only the efficacy but also the toxicity. And around 90% of kids that receive this gene therapy develop hepatitis to some degree. And there are two reasons possibly for this. So these kids, they receive an injection of an AAV vector to deliver vector genomes that contain the survival motor neuron gene. And so if you work out the amount of vector they produce, that they receive, and compare it to the number of cells in their body, they are receiving about 100 times as much vector genome as they have genomes in their own body. Okay? And a lot of that vector goes to the liver. So a lot of it is going to the wrong place. So this possibly is one of the reasons why they're getting hepatitis, on top of the fact that they're getting huge amounts of immune suppression. So this is a problem. This is far and away from a perfect therapy, and there's now groups across the world. I mean, there's certainly where I'm closely linked to, Great Ormond Street Hospital in London. There's a huge amount. There's a recognition that this is a problem, and they're trying to fix this problem. So the immune response. So there's a recognition that you have to go in early. And in fact, the UK NICE, which is the medical regulator, essentially they stopped using this vector in children older than 1 year of age. And it might be because of pre-existing immune response, or it might just be because it just doesn't work very well at that age. So the kids are treated early, but there's a huge amount of work now going in to try to find out what actually is the cause of this toxicity. But really, I mean, it's obvious. There's just vast amounts of this vector going into the liver. So.
Yeah, that's why we need that viral map.
Yes, right. Yeah, the receptor map. That's right.
So it goes to the right tissue.
Yeah.
Thank you, Simon. Thank you so much. There are a lot of students here, and they'll probably want to grab you at the networking session over there. So we really appreciate it, and I'm excited to hear about the progress and the collaborations that you're doing with people at Wits University, and I hope one day we'll be doing something to do with gene therapy. For me, hopefully HIV vaccines.
Yes, right, yes.
I mean,
this is
HIV,
very exciting, yes,
for cure agenda, but for other things as well. So we're
sure.
very excited about that. So thank you for joining us.
Thank you very much. Thank you for your time.